Good evening, my name is Mariana Nava, and I'm going to present you about maguey, Agave salmiana, micropropagation. Agave salmiana, most commonly known as maguey, is a Mexican endemic plant, which is mainly produced in the states of Hidalgo, Tlaxcala, Estado de Mexico, and Puebla. It has a medium to large size, and it has been used for textile production as well as pulque production. And it also has its own gastronomical and ornamental uses. It is a Mexican endemic plant that has the ability to adapt to stressful environmental conditions. It produces high-quality meat, and all its parts can be used, even its best. By micropropagating this species, we will be able to obtain populations with elite characteristics, as well to obtain plants with antioxidant activity. It is also important to note the key role that the Agave salmiana plays in the Sustainable Development Goal number 15, which is life on land. Maguey crops protect the land from erosion, helping it to conserve soil's nutrients. And it also captures and retains moisture that can be used by adjacent crops. Also, it is host to animals and insects, such as pollinators. Agave salmiana has some cytotoxicity activity from extracts done by using agave sap and agave leaves. We can obtain fructans and glycosides that have been shown to have a cytotoxic activity in these particular cell lines. The objective of this project is to design and implement a methodology for the in vitro micropropagation of Agave salmiana in order to obtain good quality plants to test the potential cytotoxicity activity to treat HEC293 cells, ensuring that the findings will be beneficial for our training partner, WeClone. WeClone is the first commercial micropropagation laboratory here in Mexico, in Querétaro, and it was co-founded by Dr. Ashutosh Sharma. Our hypothesis is that the treatment with a specific combination of hormones, such as IAA and BIP, will positively influence shoot induction and efficient multiplication of Agave salmiana plants in a micropropagation system. For our methodology, we had different media preparations, and we had several methodologies. For the first preparation, we only use MS as well as the hormones and agar, and its pH was adjusted to 5.7. For the second preparation, we decided to add activated charcoal to combat oxidation and two different antifungicals, Captan and Benlate. However, this will shown to be a mistake because by adding the antifungicals, the media was not able to solidify. So for the third preparation, we decided to remove the antifungicals. For the fourth methodology, which will be our final methodology, we decided to first cultivate the explants using the third methodology with activated charcoal, and then we subculture it to the first media preparation without activated charcoal. So we also had several cleaning processes. On the first methodology, we had more general cleaning process with shorter periods of time for the washes. However, for the next cleaning process that we developed, that was for the third methodology, as for the second methodology, the media did not solidify, which meant that we had no samples of Agave salmiana that were cleaned or cultivated. So for the third methodology, we decided to add a bath with antifungicals, Benlate and Captan, as well as a bath with microtin to combat the contamination of fungi and bacteria. And also, we decided to add for the fourth and final methodology an antioxidant bath. So for this final methodology, we first made the selection of the mother plant that was done by our training partner. And once we obtained the plant, we removed the leaves and its roots. And we did first a superficial wash with twin 20, and then we did a second wash also with twin 20, this time at 2% for 30 minutes. We then extracted the meristematic explants, and we had a 1 hour wash with antifungicals, 2% Benlate and 2% Captan, and 1 hour wash with microdin at 2%. We had then a wash of 35 minutes with 40% chlorine, and finally an antioxidant bath. This was done with citric acid and absorbic acid at a concentration of 150 milligrams per liter. We then removed the damaged tissue and we placed it on the first media, which was with activated charcoal, and we then subcultured it with the media without activated charcoal. For the cytotoxicity assay, we will be using HEC293 cells, which are human embryonic kidney cells. And for the extract preparation, we first wash the leaves. We dry them at 40 degrees Celsius for 8 days. We grounded them and we mix 10 milliliters of methanol with 1 gram of powder. We place it on the sonicator, we centrifuge it, and we finally filter it to sterilize it. It was stored at 4 degrees Celsius. For the cytotoxicity assay, we'll be using 4 different concentrations, as well as a positive and a negative control. And we will be reading the absorbance at 570 nanometers after 24 hours and after 48 hours. For the results, we can see that in the first methodology, we had a lot of bacteria and fungi, and we also had a lot of oxidation. For the second methodology, we were not able to use it because the media did not solidify. This was due to the fact that by adding the antifungicals, we lowered the pH from 5.7 to 2.1 after the autoclaving process. On the third methodology, we had fungi as well as oxidation, but we had one positive result, one healthy explant that had shoot formation. For the fourth methodology, we had no contamination at all, but the explants remained healthy only for 2 days after they began oxidation also. So after 4 days, we decided to subculture all of the explants of the fourth methodology. And after 13 days, we also decided to subculture the only positive result that we obtained for the third methodology. So these explants were subcultivated into MS media with only hormones. But after 6 days, all of the explants we subcultured from the fourth methodology resulted in necrosis. However, the only healthy explants from third methodology did survive. So we had a lot of bacterial contamination, and this was present in the first methodologies in all of the explants, which leads us to believe that it was endogenous bacteria. This bacteria, in the case of Agave salmiana, is Bacillus, Enterobacter, and Lachlericia. We also had fungi, but this was only present on a handful of explants, which leads us to believe that it was due to poor aseptic technique. The oxidation problem is a problem that is present in a lot of in vitro culture, and it is due to the fact of damaged tissues. These damaged tissues can be a result from an aggressive disinfection process, as well from the cuts made to the explant. So by damaging these tissues, the production of the phenolic compounds is obtained, and this production will be absorbed by the media. It will inhibit growth, and it will lead to tissue death. For the cytotoxicity assay, all of our results of 24 hours and 48 hours presented percentages of viability always above 70%, which means that the concentrations used were not cytotoxic. So if we want to furthermore research this cytotoxicity activity, we will need to use a different extract preparation, as well as different concentrations for longer times of exposure. For our statistical analysis, we wanted to test the difference in the mean of the number of shoot formation per explant in the different concentrations used, but it was not possible due to the fact that no growth was obtained, as we had a lot of problems with the cleaning process. However, ideally, if we had obtained this data, our response variable would have been the number of shoots grown per treatment, and we will have done an ANOVA of 1 factor 4 levels. For the cytotoxicity assay, we wanted to test the difference in the mean of the absorbance value read at 570 nanometers for the different concentrations used. However, this was also not possible due to the fact that we had not enough repetitions, which meant that no possible ANOVA analysis was done. So in conclusion, for the micropropagation process, we were unable to test the effectiveness of the different treatments due to the contamination and oxidation problems. But we were able to develop a cleaning method that successfully addressed the bacterial and fungal contamination. but we will need to do further research and further analysis in order to combat the oxidation problem. We will also need to expand the test with different concentrations of the extracts or with a different plant source to fully comprehend the cytotoxicity activity of Agave salmiana. And we also need to do more replicates and to continue testing in order to obtain significant data that will allow us to have a statistical analysis. That will be all. Thank you very much for your attention.